Hello dear students, welcome back to one more video session. Here I'm going to explain the stages of the interface. So what is the first stage of the interface? Gap 1 phase or stage of the interface. It takes up to 11 hours in the cell cycle phase. So here you could see a cell, that means whatever the cell which undergoes cell division, that is mitosis or meiosis, doesn't matter, they have to enter into this phase G1. And here you could see the same cell which is now into the second stage of the interface, that is called as synthetic phase or synthesis phase, or in short S phase. Okay. The last stage of the interface, the same cell now entered into the third stage and the last stage of the interface, that is called as G2 phase or gap 2 phase. And after this it will enter into M phase. It could be mitosis or meiosis. So here we study what are the things or what are the changes we'll see inside a cell when a cell enters into G1 phase. Okay, simple. First, see this cell. This cell contains a nucleus, nuclear membrane, and two mitochondria, and something is there on the right top corner. And here also you see a green color paper strip, and you could see two chalk pieces. Okay, let me relate these things to the molecular level, that is the chemicals which is produced inside the cell when a cell enters into G1 phase. The first is, see, what is this? This is nothing but the chromatin. It is nothing but DNA, correct? DNA containing histones and non-histone proteins and even scaffold proteins. Everything is there actually. So this is now the chromatin material, chromatin. Okay, nothing but DNA itself. I have used two colors. One is red and blue for the single chromatin. Very simple. You will understand it why I have used two different colors when I discuss about the mitosis phase. Okay. So these two colors are present in the single chromatin, but in real you will not see colors. Okay, the chromatin will be colorless itself. Okay, next you see T-like structures. What is this? It is nothing but nucleotides. Okay, and you see colorful structures here. This one is one amino acid, this is another amino acid, third, four, five, many amino acids are there. You could see a polypeptide chain here. These amino acids are synthesized in G1 phase. If you remember the code to remember G1 phase, what is the code? RNA is not equal to MC square. What is R here? R is, in the G1 phase, a cell produces RNA. So this is the thing. Where exactly the RNA is synthesized? The RNA is synthesized from inside the nucleus, then it comes out and enters into the cytoplasm and it will remain there. Okay, it has got some functions. So RNA is produced. Next is nucleotides. These nucleotides are produced from this chromatin, that is nothing but DNA, and you could see this is one nucleotide. This is the second nucleotide. This is the third nucleotides. These nucleotides are nothing but the basic unit of the polynucleotide chain of the DNA, which makes up the single strand of the DNA. See, if I align these nucleotides like this, you could see the strand of the DNA, and this is the nitrogenous bases. Yes, simple. Okay, so we'll scatter this like this. So nucleotides are also synthesized. Then RNA. What is A here? This particular chromatin now produces the proteins, particularly the amino acids, the basic units of the proteins, and they are linked by, these amino acids are linked by, see, this is one amino acid, this is other amino acid. They are linked by the peptide linkages, peptide bonds. So finally a polypeptide chain is produced containing many different amino acids. What is the main function of these amino acids? They are going to make up the histones. But in G1 phase, histones are not synthesized. But in the S phase, you could see the histones. Can you remember the different types of histones? There are 5 histones. H1, H2A, H2B, H3, H4. Here, this could be compared to H2A, H2B, then H3, H4. Okay, H1 is not here, doesn't matter actually. Okay, so these amino acid chain will make up these histones, that is nothing but proteins. Okay, they play an important role in the packaging of the DNA. So let me bring it inside, because they should be inside the nucleus. Okay, now E is not equal to MC square. So that means you could see one mitochondria here and other mitochondria there. That means here we have two mitochondria which will not divide further. Got it? So they remain as it is. That means there is no increase in the number of mitochondria when a cell enters into G1 phase. Remember this. Then C square. So first C is the centriole. I have used chalk pieces here. How many are there? 2 are there. That means 2 centrioles are there. The number of centrioles remain same in the cell when a cell is in G1 phase. That is 2 itself. Again, you will not see increase in the centrioles or multiplication of the centrioles in the G1 phase. Done. Okay. Next is one more C is there. That is the chloroplast. But here I have prepared all this stuff thinking that it is animal cell. So you will not see the chloroplast. But imagine instead of mitochondria, chloroplast is here. Okay. So chloroplast number also remains the same. They also will not undergo multiplication. That's it. Okay. So this is the thing. I think no need to explain the structure of this chromatin. You know what is there inside the chromatin. Nothing but the DNA itself with some proteins. It could be histone proteins or non-histone proteins or scaffold proteins. Everything will be there. Got it? If I magnify this, what you see? You see DNA. Yes, done. So this is G1 phase. How to remember G1 phase? RNA is not equal to MC square. Easy, done. Next, we go to the second stage. The same cell now is entering into the second stage of the interphase, that is synthesis phase. Here, what you see is the mitochondria number remains as it is. Okay, still you don't see the changes in the number of mitochondria, chloroplast or centrioles. The number remains same itself. But what you see is these amino acids which are produced in G1 phase now transformed or produced histone particles or histone proteins. See, these 4 are histones. Okay, they are present inside the nucleus. But here these amino acids are produced inside the cytoplasm, but after modification of these amino acids or chain of polypeptide chain, you could see the histones, they are now inside they are brought back to the nucleus. Done. Because they play an important role in the packaging of the DNA, compression of the DNA. Yes. Okay. Now, what you have to remember for S phase, the important event that takes place in S phase is the replication of the DNA. What is replication? Replication is nothing but the multiplication of the DNA. So, here, see the length of this chromatin. See this. So, it's very short actually. Okay. But still, the ploidy level of this cell is 2n. So, if I cut this into many pieces, it's going to form how many chromosomes? If I compare it to human cells, 46 chromosomes. Got it? If I cut this chromatin, it will form 46 chromosomes. Okay. Now, in the S phase, see, the length has increased. It's very, very long actually. See, it's very, very, very long. Can you see? This is long because of the replication. See, this is the earlier red color chromatin. Now it has increased its length because of the replication. Again, same thing. You could see the blue color also. It has increased its length because of the DNA replication. But still, if I cut this, I'm going to get 46 chromosome itself. That is, the number of chromosome remain the same, but length has increased. What you say now? The chromatin, earlier it was 2C. Now it has become 4C, that is double, but the chromosome number remains the same. Got it? Why? Even if I cut this into many pieces, I get only 46 chromosome. Got it? But the length is more for each chromosome. Got it? So length of the chromatin will be more for each chromosome in the S phase. Why? Because when it enters anaphase, so you know that separation of the sister chromatids takes place, making sure that each daughter cell will get equal number or equal length of the chromatin. Got it? So this is the thing. So 2C becomes 4C. Very important stage. Okay. Next is the last stage of the interphase, that is the G2 phase. Let me bring this chromatin back to this. Again, you don't see any changes in the chromatin. Okay, we'll keep it as it is. What is the code to remember? RTP is equal to MC square. Okay, what is R here? The R is nothing but again the RNAs are being synthesized. Again, the RNA could be synthesized. Okay, then T. What is T? T is nothing but the tubulin, a type of tubulin. There are two types of tubulin. One is alpha tubulin and beta tubulin will be synthesized from the same chromatin, and they will be lying inside the cytoplasm. What is the function of them? So they are very important for the formation of the spindle fibers or aster rays. Got it? So they play an important role. Okay. So if you just take the aster rays and cut it, what you see is you see the tubulin protein. Done. Okay. Next is P. What is P here? Some proteins will be synthesized. Okay. Some proteins will be synthesized. Okay. It is not mentioned here. Not required actually. The proteins are required for further process. That is for mitosis or meiosis. It doesn't matter. Next is equal to mg square. That means earlier we had how many mitochondria? 2 in each cell. That is when cell enters into G1 phase. But now 2, 3, 4. We have got 4 mitochondria. That is the mitochondria also undergoes multiplication by a simple process called as fission. So the number is increased. That is doubles. Got it? Okay. Next is C square. In that C is earlier how many centrioles we had in G1 phase? 2. But now you could see 2 pairs of centrioles. Correct? 2 pairs of the centrioles will be seen. This is very important actually. So centrioles also doubles. If it is in the case of plant cell, the mitochondria number also increases. At the same time, chloroplast number also increases. But as I said earlier that I am just taking this as animal cell. Done. So these are the events you see in the interphase. What is the duration which normally takes for G1 phase for a cell? It is 11 hours. Then for S phase, hardly 5 to 6 hours. Okay. For G2 phase, again, hardly 6 to 7 hours. Remember it, which is the longest phase of the cell in interphase, that is G1 phase. Hope you understood this. So you can check it again. If you have any confusions, you can just leave a text message. Fine. Now we will study the important phase of cell cycle and cell division, that is M phase. Here M is mitosis. So I'll be talking about mitosis, not meiosis. Okay. Mitosis is also called as equational division because the total number of chromosomes present in the two daughter cells after the completion of the mitosis will be same as of parental cell. For example, if a cell, that is parental cell, present in human being has 46 chromosomes which enters the interphase and completes mitosis, that is prophase, metaphase, anaphase, telophase. At the end of mitosis, you get two daughter cells from the parental cell and each daughter cell will contain 46 chromosomes. So it is called as equational division. Simple. Let us understand that how. Okay. Here I am going to talk about the first phase of the mitosis, that is prophase. See, this particular cell has completed the 3 important stages of the interphase, that is G1, S and G2 now entered into the first phase of mitosis, that is prophase. Okay, what is the meaning of pro? The first. Got it? Okay. Here you have to remember a trick, that is a sentence, that is cut, can, gen. What is cut? In that C is condensation of the chromatin. Okay, what is the meaning of condensation? This is condensation. Got it? Condensation. Okay, it's like nothing but compression. Okay, next is untangled. Very simple. I can easily unwind this chromatin, that is called as untangling of the chromatin. Like this. So this is untangling. Okay. Next is thickening. As the condensation is happening, it looks very thick now, correct? So thick. So thickening. So 3 events are taking place. Okay. Next is, you see, can. What is can? The centrioles. Okay. Centrioles already divided in the G2 phase. Now a pair of centrioles move towards the opposite pole. Yes. So it is moving towards the opposite pole. Done. Okay, so we have here 2 centrioles and in the opposite pole also we have 2 centrioles. Easy. Okay. Next is the formation of or production of aster rays in the case of animal cells or spindle fibers in the case of plant cells. So the centrioles now will produce the aster rays. Okay. So this is the aster rays are produced from the centrioles. Yes, can you understand this? Like this, like this, like this. It starts producing the aster rays. If it is in the case of animal cell, like this. For a good picture, yeah. This is actually the aster rays produced from the centrioles in the metaphase stage, but in the prophase itself you could see it. Okay, the centrioles now releases aster rays. So these aster rays starts moving towards the opposite pole. So from this pole, aster rays now starts moving like this to the opposite pole. Got it? Okay. Next important event is the nuclear membrane, that is CAN. Nuclear membrane starts to disappear. In the earlier stage, we had a very good nuclear membrane. It was visible, that is in the G2 phase of the interface, but now you could see they starts to disappear. The nuclear membrane starts to disappear. That doesn't mean that nucleus disappears. It will be there, but nuclear membrane disappears. That's it. Why? To provide space for the movement of the chromosome. That's it. Okay. Next is gen. Cut CAN is done. Now gen. What is G here? The Golgi. The Golgi or Golgi apparatus starts to disappear. So you cannot see the Golgi. Okay, we'll keep it aside. Done. Next is E, that is endoplasmic reticulum. This endoplasmic reticulum also cannot be seen. It disappears. Next is the nucleolus. This is nucleolus now. It also disappears. Gone. That's it. Got it? So this is all the events taking place in the prophase. Done. Now the cell is entering into another stage that is called as metaphase. Before that, let me just clear, when the condensation, untangling and thickening of the chromosome or chromatin is taking place, it will form a structure that is called as chromosome. So formation of the chromosome normally takes place in the late prophase, that is just before it is entering into the M phase or metaphase. Got it? So you see in the chromatin I have used two different colors, that is red and blue here. Yes, now they have formed a wonderful X-shaped structures, that is nothing but called as chromosome. So chromosomes are formed because of the condensation, untangling and thickening of the chromatin in the late prophase. Got it? Late prophase. Don't write like in the metaphase chromosomes are produced. No, in the late prophase the chromosomes are seen, X-shaped structure. The next stage is the metaphase, that is the second phase of the mitosis. So we keep the red chromosome here and the blue chromosome here. That's it. Okay. So they are actually separated. And these two chromosomes are not homologous chromosome. Please understand that. What is the meaning of non-homologous and homologous chromosome? Homologous chromosomes are nothing but when this particular chromosome has a copy exactly similar. You can imagine twins. Okay, exactly similar to each other. So that is homologous. Morphologically and genetically same, they are homologous chromosome. So for this is not homologous. So these two are now non-homologous chromosome. I'll make you understand it later. Okay, so please understand. Genetically and morphologically both are different. For example, let us see morphologically. Are they same in their structure? No. One is bit fat, the other one is lean. Yes, that's it. Simple. Okay, so they are non-homologous chromosome. Okay, these two non-homologous chromosome actually normal like present inside the cell because we have a space, nuclear membrane is not there, so the chromosomes can align at anywhere else. Now the aster rays which are produced from the centrioles, okay, they starts moving. Okay, they starts moving. Some aster rays move towards the opposite pole. You could see here this particular centriole released this aster ray which touched the opposite pole. Okay, that means it did not come across with the chromosome, but some aster rays here which is released from the centriole come across with some chromosomes here. In this case it has come across with this particular blue color chromosome. Aster rays, okay, come across with this particular red color chromosome. Got it? Okay, so particularly the aster rays, when it binds to the kinetochore of the chromosome. What is kinetochore? It's a type of protein. How many kinetochores will be there in a chromosome? Total 2. One is here, the other one is exactly opposite to that. That is in the opposite side. Okay, that is exactly adjacent to the centromere of the chromosome. That's it. Okay, so one aster ray now touches the kinetochore and binds to it. this asterase which is produced from another set of centrioles which is now going to touch the kinetochore. For example, let me just show you that. So let us imagine this asterase now touches the kinetochore. Okay, here also same. This asterase now binds to the kinetochore of the chromosome. Okay, so done. We'll keep this. Now the asterase pushes or pulls. See, in this case it is pushes and in this case it pulls the chromosome towards the center, exactly towards the equator. Okay, so now both chromosomes align at the equator of the cell. Yes, so this structure or alignment of the chromosomes at the equator of the cell is called as metaphase plate. This is seen only in metaphase. Done. Easy. Okay, now we'll see what is the fate of these chromosomes after they align in the metaphase stage. That means they are now ready to enter into the third phase of the mitosis, that is called as anaphase. Okay, here students, you need to understand how exactly chromosomes now separate. Got it? There is a clear-cut difference between anaphase of mitosis and anaphase 1 of meiosis 1. In the case of anaphase 1 of meiosis 1, this entire chromosome moves towards the opposite pole like this. But this is not meiosis, this is mitosis. Okay, so here you need to understand it properly. Okay, now in the metaphase stage, the chromosomes are aligned at the equator. Now the centromere breaks. Okay, you could see here it's already broken. And now the asterase, I think no need to explain it again. The asterase now starts to pull one sister chromatids towards this particular end of the cell from each chromosomes. Got it? Okay, so this one is pulled to the opposite pole by the asterase released from this particular centriole. So like this. Okay, now two sister chromatids are now separated. Got it? Okay, so this is the thing now. Okay, this is anaphase. Got it? Okay, for anaphase to be initiated, we need one complex called as anaphase promoting complex. I will explain it in the next class. Okay, don't worry about it. So this is the thing happens in anaphase. What is important here to remember? The centromere break from the chromosome and each sister chromatids now starts to move towards the opposite pole like this. Done? Okay. The last phase of the mitosis is the telophase. Telo means terminal. Here decondensation of the chromatin takes place. So decondensation has occurred. Correct? Okay, no condensation. The chromatin is entirely free. Here also decondensation. Yes, that means you don't see the chromosome. That means this particular chromosome, after they move into the pole, that is opposite poles, they form a structure like this. This is now chromatin. Got it? You don't see X-shaped structure here. Okay, decondensation is seen. Then tangling will be there. Tangling means it's bit they tangle like this. It's very difficult to untangle them now. Okay, then it becomes very thin now. Thin like this. Okay, so these are the characteristics you see in the telophase. Done. Okay, then can, that is centrioles. Here in this case, do you see centrioles? No, centrioles are gone now. It's not there. Centrioles now starts to disappear. Okay, then asterase. Can you see the asterase? No, centrioles are already disappeared. Even the asterase also already disappeared. Got it? Okay, then we will just bring some structures from here. You see Golgi. Now it is returned back. Okay, then nucleolus. I have got only one plate. Okay, let us just put this. This is now nucleolus. Okay, nucleolus now starts to reappear inside each nucleus. Then you see this endoplasmic reticulum also reappears, and also here also. Okay, so everything returns back. So this is the thing. Got it? Okay, now the important thing is cytokinesis. So far we have discussed about karyokinesis. What is karyokinesis? Nothing but the division of the chromosome. That is movement of the chromosome. Okay, so that is karyokinesis. The meaning of karyo means chromosome. Okay, kinesis means division. Done? Easy? Okay. The last stage of mitosis, that is after telophase, is the cytokinesis. What is cytokinesis? Cytokinesis is the division of the cell to form two daughter cells. Now, one cell containing two nuclei, that is two nucleus, correct? Okay. So, here, if it is animal cell, you see a cleavage furrow. What is cleavage furrow? See this. Now the cell membrane starts to move towards the center like this. This is cleavage furrow. Okay, so this also, this cell membrane now starts to move towards the center like this. Like this. Done. So now this part now separates from this part. So two new cells, two daughter cells. It is seen only in animal cells. That is formation of a cleavage furrow because of the movement of the cell membrane towards the center. Okay, we'll just compare this to the plant cell. In the plant cell, you don't see the cleavage furrow, but instead of that, you see a cell wall which is now formed exactly at the center of the cell, separating the two daughter cells like this. Can you see now? Yes. So this is called as cell plate. Cell plate formed because of the cell wall itself. So the cell wall produces a small structure which in future it's going to be cell wall of this cell, at the same time cell wall of this new cell also. So this is all about the cytokinesis. Done, easy. Hope you understood this session. Thank you.